Functional biological nanostructures have been the inspiration for many different types of nanostructured material, and I'm going to end with this topic of biomimicry by addressing just one aspect of this, which is the use of specific proteins to assemble intricate nanoscale structures. The images here in this panel labeled A are of abalone shell, and in B, the mineral skeleton of a diatom. The abalone shell has some remarkable properties. The mineral composes most of the material, so you see here these crystalline sheets of mineral held together by little pillars, most of which is again mineral, but a significant protein fraction in these pillars. A small percentage of protein material in the overall shell composition results in a shell that is much more resistant to impact by many orders of magnitude than the mineral material alone. So a tiny amount of protein completely dominates the mechanical properties of the shell and appears to guide its self-assembly. Look at the intricate detail in the self-assembly of the skeleton of a diatom, once again somehow directed specifically by a very small fraction of protein component associated with the mineral deposits. You, of course, will recognize biomineralization in bone and tooth and so on in higher organisms, and the question of exactly how this self-assembly occurs is still a subject of research. But in researching something like an abalone shell, it was found that specific regions of the protein would bind very specifically to certain mineral faces, thus orienting them in an absolute crystallographic direction. How is this done? We will discuss the structure of proteins in the next chapter and the way in which their sequence is directed by DNA, but except for now that a gene of DNA with the 4 bases A, T, C, and G arranged in some order codes for the residues on the backbone of a protein. There are 20 such residues, and so the code on the DNA then gets translated to the composition of a protein. It was found that very specific residue sequences in a relatively small number could form incredibly specific molecular glues to hold mineral components in place. This has led to a whole new field of research in which biologically generated polymers, peptides, i.e. small runs of protein, are used as incredibly specific surface glues to hold inorganic materials together. How is this done? It's done by a process of random combinatorial selection, which I will explain now. So the first thing that's done is to create a sequence of DNA, which may have some fixed sequence at the top, but then in a region here have inserted a random library. This may be 40 or so bases of DNA, in which instead of being some fixed sequence ATCG, is completely random. This is then followed up again by some fixed determined sequence, and we'll talk in a minute about what these end pieces do. This DNA is synthesized and inserted into the genes of a little organism called a bacteriophage. How does one make random DNA? Actually, it's simpler than making DNA of a fixed composition. When one gets to this point in the DNA polymer, on the DNA synthesizer machine, one simply turns on the spigots for all 4 bases, A, T, C, and G, and runs through the acid cycles that allow these bases to be deprotected and added to the chain, but adding now quite randomly, depending on what happens to be near to the chain at each point, for each chain on the bead on which the DNA is synthesized in A, T, or C, or G, with the result that one then forms some random library of sequence, and then finally terminates the chain by then adding just an A, or just a C, or just a T, or just a G at each of the following steps. When this is done, standard molecular biology techniques can then be used to splice this piece of DNA into the genome of a bug called a bacteriophage. So this is a rather simple organism which contains its genome inside a protein package. And the splicing is arranged so that specifically the proteins that appear on the head of the bacteriophage will now express some random loop of peptides as a result of the incorporation of this piece of artificial DNA into their genome. So this means that if you culture a dish containing 1 billion different bacteriophage, they will present at least 1 billion different peptides at their heads. The bacteriophage are then incubated with the surface of interest. I believe for this particular image here, the substrate was indium phosphide, a semiconductor, and more specifically, the 1,0,0 face of indium phosphide. So the bacteria are deposited onto the surface, and only those bacteriophage with sequences specifically sticky to the surface remain on it. The surface is then rinsed, the few remaining bacteriophage are recovered, their DNA is extracted and multiplied up by a process called a polymerase chain reaction. The sequence is found that bind, and these are then used to make potential candidate peptide glues. One can go through this sequence a number of times, for example, then taking the viruses, bacteriophage that bound to the surface, and incubating them specifically with another surface that you don't want them to stick to, like say indium phosphide 110, and then rinsing that and collecting now what comes off when you rinse rather than what sticks, and going through the cycle a few times to get highly specific sequences. The images here show filamentous bacteriophage sticking to this surface during one of the selection cycles. By processes like this, glues can be made that are specific at one portion, let's say for a silicon 100 surface, and at another portion for a specific surface of a magnetic nanoparticle. And so these things then act as molecular assembly factors for making complex structures. We end this section with one more illustration of mimicking biology. In this case, a little animal called a brittle star that, although it has no central nervous system and no obvious eye, is known to be light sensitive. It was found that its biomineralization formed a dense array of nanolenses, and these are what collected light for the brittle star. Arrays of nanolenses like this have been fabricated and have found use in adaptive optics.